My name is Sam Gascoigne, and thank you for joining me virtually today to hear about a paper that we published just a few months ago titled A Standard Protocol to Report Discrete Stage Structured Demographic Information. Today I'm going to be running through the paper and specifically talking about why we wrote the paper, the story of it leading up to the paper, why we put in the paper what we did, and where we see MPM communication going forward in the years to come. And just before we kick off, I'd like to start with some preemptive thanks. This paper was a massive slog. It took quite a while to do, including 5 undergrads, 5 postgrads, 8 ECRs, 22 PIs across 8 countries and 33 institutions. It was a massive effort on many parts, not just my own. So I'd like to say massive thank you to all those. And secondly, to Natalie and Maya and Methods in Ecology and Evolution for getting this set up for us. Now, just before we begin, I realized the star of the show is something called a matrix population model. And I realize some on this call might not be aware of what these are, so you might be quite agnostic to the star of the show. So what I'm going to quickly do is run over what a matrix population model is, why we use them, and how they've impacted population ecology. So let's take a step back. Imagine you're a young budding ecologist, population ecologist, interested in studying your population of interest. In this situation, because we're allowing our imaginations to run wild, imagine it's a Pokemon population in the clouds. Specifically, you're interested in whether your population of interest is increasing over time, staying the same, or decreasing. Across demography, we have lots of ways of modeling populations. Some of those methods are called structured demographic models. And some of those structured demographic models allow us to take in the individual heterogeneity we see across a population and add those as components of our model. For example, if we look at our Pokemon population, we see that there are different types. Some of them bigger, some of them are smaller. If you're interested in birds, you might have juveniles and adults. If you're interested in insects, you might have different instars. But for this sake, we're thinking of Pokemon. As we model the population over time, we can construct censuses and calculate the per capita contributions of individuals to different individuals' time. Now, that is a bit complicated, but we can visualize this very simply with something that's called the life cycle graph. Here, we've taken the different stages, different classes within our Pokemon population, and with these arrows, we're able to show the per capita contribution of individuals in the first stage to the second stage by the value A, from the second to the third by the value B, and we're also able to show reproductive components, for example, from the final stage to the first stage as C. Matrix population models are especially handy, especially if you're teaching demographic methods, because the life cycle graph is intimately connected to the matrix you end up constructing. For example, if we're modeling this Pokemon population, the matrix that we would construct would be a 3x3 matrix population model. Now, if we imagine the columns represent the stage at time t, and the rows represent the stage at time t plus 1, so at the end of the arrows, we can very easily add the values we calculate for our Pokemon population in the sky and build a matrix population model. That's all a matrix population model is. So what I'm going to be talking about are these mathematical structures. Now, in addition to being interesting and fun to talk about, they've been extremely useful in population ecology. Anything from comparative demography to life history theory to resilience dynamics to conservation practices. MPMs represent a very useful tool in population ecology. Now, to support this effort, there have been databases around. Specifically, the databases I'm going to be talking about today are the Compadre and Comadre databases. These are 2 sister databases that we support, where Compadre is a database of MPMs associated with plant species, plant populations, and Comadre is for animals. And we have lots of species. So in the current issue, you can see on the website, we have 792 species in Compadre and 429 in Comadre. Within this, just if this is of interest to anyone online, we also have lots of open access user guidelines education materials. So if you're interested in using MPMs in the classroom, in your research, please do feel free to go to the education tab on the resources tab at the top of the website. However, beyond communication, we're really talking about research here. We're talking about the use of MPMs in population ecology research. To create the best tool for you, the researchers, we have two packages that are intimately connected to each other that allow you from R to access the databases, import the data, screen the data, and create some inferences. For example, we've got the R compadre package, which allows you to import the newest version of the databases that you're working with, do some data cleaning. It's all integrated with tidyverse. And secondly, if you're interested in calculating, let's say, life history traits, there is a package also called RAGE that works with these data objects for whatever inferences you want to draw from matrix population models. And just to give you a sense of what these databases are that we're going to be talking about, we have reasonable, although biased, but reasonable geographic coverage. Well, all of these points are not all the MPMs in the databases. As we will talk about later, not all MPMs are given with a latitude and longitude coordinate. However, those that we do, we're able to show that we have reasonable coverage across the globe in some plants, not as much in animals. But yeah, if any of these regions or any species within these areas of interest to use, please do feel free to reach out to us via Twitter, email, or just over email. And we can talk about how we can use the databases to power your research. So now that we've run over what the databases are, we're now going to be talking about behind the scenes, because it's really behind the scenes, which is the reason we wrote this paper we're going to be talking about today. So what do we do? What does the compadre and comadre teams do? First, we have little alerts that tell us when MPM-related publications are published. We take these publications. We do some standardizing and error checking of these papers to see how we can get the data out to you all. And last but not least, we use packages, we do workshops to make sure that the data are being used to empower your research. That is the simple model of what we do. However, some issues have thrown a cog in the works. First, MPMs are becoming really, really popular. This graph is a bit out of date, but what we've seen in terms of the publication of MPMs is more and more people are using them. This is good. They're a very useful tool, but it also means when what we need to integrate into the databases is becoming more and more. And what is exacerbating that problem is that there are some common issues. Firstly, MPM-related papers do not always report the MPMs they use. So we'll get flagged as a paper that we need to digitize to try and get it out to you all to empower your research, but the MPM won't be there. It won't be there in the supplementary materials either. Secondly, sometimes the methods are not properly communicated, or the metadata, for example, latitude and longitude, census duration, sometimes those really important aspects of the study are not communicated with the demographic rates they're being attributed to. And last but not least, unfortunately, there are some errors that sometimes arise. This happens, but unfortunately, there are some common errors. If you're interested in looking into the errors that do happen, I'd highly recommend this paper by Bruce Kendall. And he notes that there are actually some very simple, avoidable errors that are commonly being made in MPM construction. So this is where we're at, where we've got an increased number of supply of MPMs, and we've got a rate limiting step. We've got issues in terms of how we can get these data to power your research. And it's not just us at Compadre and Comadre. All of these MPMs are either open access or within the publications. And we're of interest in trying to get the best quality data for you in your studies. So we noted these issues, and we were left with a problem. It's like, is this just what we're seeing, or is this not a true representation of the current state of affairs in MPM communication? To get this sense, we chose two avenues. First, we did a screen of 300 papers. This included 150 papers from Comadre and 150 papers from Compadre. And secondly, we did a survey of experts in structured demography. First, I'm going to talk about the screen, and this is now where we're getting into talking about the paper. Within the screen, we looked at each paper that we screened, and these papers, I should say, were already digitized in Compadre and Comadre. So this means that one of our research assistants, we call them compadrinos within the group. One of the compadrinos must have worked very hard to get them digitized if certain data were missing. So whilst all of the papers that were reviewed, that were screened, had been data collected, had been screened, that's not necessarily meaning that they were all easy to digitize. So across these 300 papers, across the two databases, we found that some aspects of MPM communication are well described, like the location, which would be the city or the country. The MPM themselves are quite well communicated. Census duration, vital rate formulas are also well communicated. But I realize I say that, and it might not ring a bell. It's like, Sam, I thought you were talking about matrices. Why are we talking about formulas? The reason is matrices are often constructed from underlying components. So a matrix often has submatrices that build them up. For example, we have this figure in the paper. Imagine a matrix population model here noted as A. Some of the demographic rates have contributions from only one demographic rate. For example, stasis from stage 1 to stage 1, which would be the top left-hand corner of matrix A, only has one demographic rate that builds it up. However, the contributions from stage 2 to stage 1, which is second column, first row, has two demographic rates that build it up. Specifically, a retrogression or a shrinkage rate that you can see in U, and a sexual reproduction component. Now, if you are interested in calculating any life history traits such as life generation time, reproductive output, you need to know what part of the values in A are attributed to reproduction and which are not. So this is very important. It's from the screen, it was reasonably reported, but I just hope that clarifies what we mean by vital rate formulas, because we'll be talking about those in a bit as well. Also, projection interval was reasonably reported, but there were some aspects that weren't, specifically latitude and longitude coordinates, life cycle graphs that we talked about with the Pokemon population, and last but not least, population vectors. These are vectors containing the number of individuals in each stage. So that was from the screen. But we realized after doing the screen that not all components of MPM communication are made equally. For example, the location is less important than the MPM. The MPM is what we actually need, whether it's in Beirut or London, is not necessarily stopping the vector multiplication. So what we then did using these screen data is we started categorizing papers into how well they communicate the components that matter most. And we did this all the way from inadequate papers, so papers that didn't communicate the MPM used, in yellow, all the way to the gold standard in purple. Each of the intermediates are adding layers. So the MPM plus VR is MPM plus the demographic rate decomposition. Plus POP means adding the population vector. Plus eco means having the latitude and longitude coordinates. Whereas what we always want is for all the papers to be in this purple band. But what really shocked us is that less than 16% across both databases are reaching this gold standard of MPM communication. So that was the first avenue for us to get a state of the current affairs. And admittedly, this was quite eye-opening, because it means that what we were seeing on the front lines of getting and digitizing papers is actually also what are currently in the databases. The second way that we did this is we reached out to experts in structured demography. So these are all experts that have published papers as corresponding or first authors using data where they've constructed matrix population models. And what we did is the exact same thing that we did with the screen, asking researchers if they noted that certain components of MPM communication were well reported. So whether they strongly agree that papers are doing a good job of communicating trait names, census duration, projection interval, and so on, or whether they're doing a poor job, and that would be strongly disagree. And what we found here is, again, another gradient. Some aspects of MPM communication researchers believe are being well reported, like trait names. So the descriptions of categories within the structured population model, whereas others are not. So the vital rate formulas that we were just talking about, researchers believe we're not doing a very good job at communicating them. Now, at the end of this survey, we had an idea what was happening. We had an idea that we're not doing a good job within the field at large. So we had a question at the end where researchers were asked, do you think population ecology would benefit from a standardized method for communicating MPMs? As you can see here at the bottom in blue, 83% of researchers agree that we would benefit from a standard method of communicating matrix population models. So this gave us a sense, these two, the screen and the survey, gave us a sense of that we're not doing the best job within structured demography of communicating the methods and the data that we use to build our MPMs. And this left us with a bit of a pickle, really, because we were wanting to actually provide the best value for the field. And we didn't necessarily know immediately how to do that. Would this be writing a blog on the website? Would this be just creating a massive thread on Twitter? And what we ended up coming to is first building a standard protocol, and second, creating a one-stop shop for researchers. If that is a miss on you, it's basically we wanted to create a body of work where you could go to it and you could say, all right, these are the common errors, this is how to fix them, and these are common methods in matrix population model construction that I might not have used, but I might want to use in the future. And this is essentially the bulk of the paper that we're talking about today. First, the standard protocol talks about every aspect of matrix population models in terms of their communication that should almost always be communicated. First is describing what type of matrix are you communicating. Now, this may seem trivial. You would have thought it would just be a square matrix. All square matrices are the same. Unfortunately, they're not. Some matrices are multidimensional. So these could involve age along with stage, along with size. So multiple aspects of structure within the population. Others may be density independent. Some might be density dependent. No matter what, this must be communicated with a matrix population model. Secondly, the life cycle diagram. The life cycle diagram, similar to what we talked about with the Pokemon population, this also would be very nice, and we would highly recommend communicating this with your MPMs. There are some times where this is a bit harder. So if you're building high dimensional matrix population models, sometimes this isn't necessarily the easiest way to do it. There are ways around it, so do feel free to reach out if you're dealing with this problem. But life cycle diagrams are very helpful when it comes to reviewers reviewing your papers, authors collaborating with you, and handling editors dealing with your papers at the review stage. In addition to these components, we also have methodological components. First being census description. So you have to say to a minimal degree when you started and when you ended your data collection, where you performed your data collection, the projection interval, so these are discrete time population models, what time interval, what length of time interval is associated with your matrix population model, the mode of reproduction, whether it's birth pulse, birth flow, and the census time, pre or post reproductive census. If any of these words that I'm saying right now you're not quite sure what they are all described in detail in the paper. The next aspects are fairly trivial, but you would be surprised they're very rarely communicated. First is describing the stages. So in the population model we talked about with the Pokemon, it was very obvious what they were, but that's because I showed you a picture showing the individual heterogeneity in the population. When we're, as population ecologists, reading papers, we don't always see your species. So it's very important to describe what construction dimensions of your matrix population models. And last but not least, vital rate definitions. Are you talking about fecundity? Are you talking about fertility? Are you talking about survival, growth, shrinkage, retrogression, clonal reproduction, sexual reproduction? All of these things need to be defined. And last but not least, we also recommend, and within the standard protocol, that you communicate the vital rate values, so the actual values that you calculate, along with standard errors and the sample sizes you use to calculate them. The matrix formula, so not just the values in the matrix, but the underlying vital rates that are used to construct them. You can see this here in the example in G, where it's multiple demographic vital rates that are being multiplied together to construct one demographic rate in the matrix population model. And last but not least, if you can construct the population vector, we recommend constructing it and communicating it as well. So this was the standard protocol. We were always going to do this given the survey results. But in addition to this, we wanted to create the one-stop shop that I mentioned. And this is a weird paper. It was a fun paper to write, but it was a weird paper to write because our intended audience isn't just researchers. It's also researchers when they're being reviewers of papers. It's also handling editors and associate editors when they're handling these papers and thinking of how it's best to communicate the methods associated with this modeling practice. So what we did is we effectively threw the kitchen sink at it. Within the paper, you're going to find an awful lot of information. Within the paper, we talk about common issues. This builds off Bruce Kendall's paper, but also there's a lot of explaining. And with the common issues, with all these common issues that you see, from census type all the way to incorrectly parameterizing fertility, and last but not least, more assumption level issues with irreducibility and ergodicity, we give recommendations for how to deal with these issues if these arise in your MPM construction. After talking about these issues, we then talk about how to best fully represent your MPM. We have the box in the paper that we just talked about, the standard protocol, but we also say why. Why is it really useful for you to do this? Why is it useful to you to follow this standard protocol? And in the future, are there ways that you can archive? And we connect this to the databases, but we also show that this can be done through other means like Zenodo and Triad. And last but not least, we're here to empower you in this paper. And specifically, there are aspects of matrix population model construction that we think would be useful to researchers that we don't see used as much. These include nonlinear dynamics, such as density dependence, frequency dependence, environment dependence. So if you're interested in variable environments or population dynamics, we have a section on this. And last but not least, we talk about multi-state models. These are extremely useful models where a lot of work has been done. And this essentially means if you have more than one axis of variation in your population model. So if you have age and stage, structuring your matrix population model, you can do an awful lot of inferences there. Now you might say, Sam, this is all well and good, but are there any examples that you use or we can look upon to guide us in writing our papers to follow this standard protocol? Yes. And we have that in the supplementary materials of the paper. We have an entire supplementary table where we talk about the data that is worth communicating, like GPS coordinates, life cycle diagrams. And we talk about the rationale and along with examples with species across the tree of life. So in addition to the manuscript proper being a vital resource, this table itself also shows you like there are many ways of communicating information, and these are some of the papers that you can use when you're constructing and communicating your matrix population models. And that is a run through of the paper. It's not exactly a paper with a narrative, but it's meant to be a resource to population ecologists and those interested in learning how they can use matrix population models to power their research. It's connected to the Compadre and Comadre databases, but if you're interested in empowering your research with these databases or with using the education materials that we use, please do feel free to reach out. And just to end on a closing point, I mentioned earlier that this is a resource for the field. We're really not trying to say this is the standard protocol because every MPM is different, but a set of guidelines. And we were hoping to spur some conversation, some progress in the field with regard to MPM communication. And what we're really happy about is after the publication of our paper, a paper by a collaborator of ours who's really been interested in uncertainty with regard to ecological models and specifically structured population models, has put on and submitted a preprint talking about how uncertainty within MPMs changes the inferences we make on the back end. So what we've talked about today, MPMs are very useful for inference. And within the standard protocol, we talk about errors being communicated as well, along with the estimates they're associated with. Emily and Owen in their work have done a really, really good job showing the impact of uncertainty and why we need to be doing a better job. They make some specific points as to how they build on our protocol, but I'm not going to steal their thunder here. Emily's been gracious enough to let me link the paper to her preprint, but I'm happy to say we are moving forward in how we're hopefully communicating MPMs, and hopefully in the future, we'll be connecting our protocol to more estimates of uncertainty as well. And I'd like to say thank you very much. Thank you everyone who's joined me online now and whoever's watching this in the future on YouTube. And I'm happy to field any questions.